Dear students, welcome to Extra Edge Rapid Revision for INI-CET exam. Even though the NEET PG 2021 exam has been postponed, CET exam, I strongly believe that your INI-CET exam will happen on the scheduled date. Because tomorrow the DM entrance exam of AIIMS is going to happen. That is for sure. Okay. If the DM entrance exam is important, MD-MS exams are equally important. Okay. So I'm sure that your exams will happen. So prepare for your INI-CET. Right. Guys, I hope you are all able to hear me. Hi Moksh Sharma. Hi Nitish Kumar. Good evening. Good evening. Right. So let's begin. Try to answer the first question. Human somatic cells, which are grown in a culture medium, die after a certain number of division. This is due to absence of which of the following enzymes? Tell me the answer. Excellent Nitish. Excellent Moksha. Good job. So somatic cells dying after certain divisions. So this is known as what? What is the name of this? You must be knowing, right? This is known as your afflix limit. Okay. So with successive cell divisions, the end portions of the linear chromosomes, right? The end portions of the linear chromosomes, they get shortened. So the answer is what? Telomerase. Excellent, 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 guys. Answer for this question is telomerase. Absence of telomerase is the reason for replicative senescence and aging. Telomerase is present in your stem cells and cancer cells. Next question. All of the following are enzymes involved in gluconeogenesis except. Tell me which enzyme is not involved in gluconeogenesis. Let us see. Okay, Nitish is saying D, Hitesh is saying B. Moksha is saying B. Nitish, why did you remove your answer? Because two people are saying option B, you now lost your confidence. Nitish, you should not lose your confidence. Right. There are three enzymes which are unique to glycolysis. So there are three irreversible steps of glycolysis. Okay. There are four enzymes which are unique for gluconeogenesis. Got my point? Look at pyruvate kinase. This is an irreversible enzyme. Irreversible step catalyzing enzyme. So we simply say irreversible enzyme. That means it's an enzyme which catalyzes an irreversible step of a metabolic reaction. Clear? Phosphoenol pyruvate to pyruvate conversion is catalyzed by your pyruvate kinase. That's production of ATP at the substrate level. So this is an irreversible step catalyzing enzyme. So this step, right, it cannot be reversed in gluconeogenesis. You don't have an enzyme that will directly convert even pyruvate to phosphoenol pyruvate. Okay, so this conversion happens by two enzymes, pyruvate carboxylase and phosphoenol pyruvate carboxykinase. Got it? Whereas enolase, right? Enolase, which is an enzyme of glycolysis, that is reversible. Are you understanding? So that's a reversible step catalyzing enzyme. So obviously that enzyme can be utilized in gluconeogenesis also. Phosphoglycerate kinase, this is also an enzyme that catalyzes reversible step. So that can also be utilized in gluconeogenesis. Pyruvate carboxylase is an unique enzyme of your gluconeogenesis. Answer the next one. Hitesh, I hope you got it right. You got the answer now. If there is any doubt, feel free to ask me. I'll try my best to explain. All of the following are key gluconeogenic enzymes except. Moksha also removed the answer. Nitish also removed the answer. Acha, now both are giving D. So key gluconeogenic enzyme means they must be involved in only gluconeogenesis. They should not be involved in glycolysis. Got my point? So key gluconeogenic enzymes means they are involved in gluconeogenesis but not in glycolysis. So the answer is phosphoglycerate kinase. Because this enzyme is involved both in glycolysis and gluconeogenesis. Okay, so it is not a key gluconeogenic enzyme. Got my point? Right. So guys, 19th April. So that is on Monday. So from Monday onwards, we are going to start a new batch in the Plus platform of Unacademy. So this is a FMGE test and analysis. So obviously FMGE means it will be useful for your NEET PG as well as INI-CET. So this FMGE test and analysis batch, it starts on 19th April. So you will have 19 subject-wide tests followed by everyday solution discussion sessions. So on 19th itself, I have a class. So the first session will be biochemistry, right? So initially we are going to start with biochemistry, then the other subjects will be followed. Tomorrow, elsewhere global challenge start. USMLE questions are integrated. Good Nitish, do it well. Okay. And we are also starting target next 2022 batch. Right. Next question. So some researcher is trying to induce insulin resistance. So what is the meaning of insulin resistance? You have insulin, but you are not getting the effect of insulin. So that means insulin is there. Okay. So after that, what happens? It has to bind to the receptor. Then there should be signal transduction. So insulin resistance means insulin is binding to the receptor, but after that there is no downstream effect. So there is resistant to the action of insulin. Right. So Nitish, Mox, Karthik. So all of you are giving correct answer. Yes. To induce insulin resistant, which you have to inhibit, which is the downstream effect of insulin receptor. When insulin binds insulin receptor, it undergoes dimerization, right? So insulin receptor actually it cross phosphorylates, right? They cross phosphorylate. Insulin receptor has intrinsic tyrosine kinase activity. So option E is the answer. Excellent, guys. So you are all giving correct answers. Next one. Will be a bit lengthy question. This is an immunology question. You know, in AIIMS, in the first year itself, we teach them immunology. So in biochemistry, you will also get immunology. Excellent, Moksh. Try to answer. Karthik Sharma is saying option D. Okay, option D, it may be, I may be hiding the option D. Option D is lack of NADPH oxidase. Okay. Okay, it is NADPH oxidase. Okay. Right. Try to answer. Acha, Nitish, you have changed it from option D to option C now. Hmm? Okay, let's analyze the question. Let me try to help you. So, a boy, he's experiencing these infections in the first year of life itself. Hmm? So, he is having diarrhea also. Look at this. He is having some kind of immunodeficiency. That is for sure. Serum calcium levels are normal. Look at the clue. So the USMLE type of questions, right? The beauty is that there will be lot of clue. There are ample amount of clues will be there. If serum calcium levels are normal, you cannot go for the option B. Failure of the third and fourth pharyngeal pouches to descend. So you rule out. Excellent, Arjun Raj. Rule out option B. Because what is this option B is saying? What is this option B is saying? Tell me. Option B. Which disease? Third and fourth. Excellent. Dijard syndrome. Excellent, Arjun. Karthik Sharma. So we rule out Dijard syndrome. Right. Next. Look at the other things. Serum calcium levels are normal and the patient's white blood cells change nitro blue tetrasolium from clear to bright blue. So there is something called NBT assay. Nitro blue tetrasolium, right? So this dye is converted into blue. That means your NADPH oxidase is present. Are you getting my point? Only in the presence of NADPH oxidase, this NBT will be converted into blue color. So you can exclude chronic granulomatous disease. Clear? So we exclude option D also. So Sankeet, so we have excluded option D also. Right. Now, however, his B and T lymphocyte counts are very low. So both B and T both are low. So what is this? B and T both low. So what will we call it as? So this is combined immunodeficiency. Okay. Arjun Raj, excellent. Moksh Sharma, excellent. So this is severe combined immunodeficiency. deficiency, SEID. This SEID is a phenotype. So SEID can be due to multiple causes. Adenosine deaminase deficiency, right? ADA deficiency. There are many other causes. So one of the causes is that interleukin 2 receptor gamma chain defect. So interleukin 2 receptors gamma chain. So that is a high affinity interleukin interleukin 2 receptor. Okay. So option A is the answer. What about option C? Inability of helper T lymphocytes to switch classes. What is this? Will helper T lymphocytes switch class? Which will switch class? Like it is your plasma cells, right? Plasma cells, they switch class. It's not your helper T lymphocytes will switch the class, right? Getting my point? So it will be your plasma cells which will switch the immunoglobulin heavy chain class. Light chain class is not going to be switched. It is the heavy chain class which will be switched. Right. So I hope you understood this clearly. So we can go to the next question. So this is one previous year question, INI-CET question only. DNA methylation is involved in all of the following except. Tell me. Moksh is thinking it is option A. Let's see. Deepan is saying it is option B. Abdul is saying it is splicing. That is option B only. Good. Remember methylation of DNA, right? That mutes. Methylation of DNA mutes the gene expression. So methylation, it mutes gene expression. So okay. So let me change option D. Option D is mismatch repair now. So it is not base excision repair. So it is mismatch repair. Okay. Sankirt, consider my change in option, then now answer. So methylation will convert the genome into a heterochromatin state. So it favors heterochromatin state. Okay. So that means it's going to silence the gene. So it's going to help in remodeling the chromatin also. So both. Okay. So gene silencing as well as chromatin remodeling. So the chromatin remodeling means, so the histone proteins are moved in such a way that the DNA is not accessible to the replication and transcription machinery. Okay. Mismatch repair. Mismatch repair is due to what? It is due to methyl, right? Methyl directed mismatch repair. So in mismatch repair, obviously your DNA methylation is involved. What about splicing? See, splicing is happening in what? It's happening in RNA. DNA methylation has nothing to do with RNA splicing, right? DNA methylation happens in DNA, not in your RNA. How can the DNA methylation affect the RNA splicing? Okay. RNA undergoes methylation, that is a different story. But here, they are specifically asking DNA methylation. Okay. So let me tell you, now we have 34,000 carefully curated questions for PG entrance exam in the Unacademy platform. To continue your practice, we are helping you. We are trying to help you. Good. Good, Moksh Sharma. I'm glad that now you understood. Try to answer the next question. For gluconeogenesis, which of the following is an effective stimulus? Tell me. Alright. Again, I think I'm hiding option D. Let me just shift the options per. Tell me, what is the answer? Sankeet is saying B. Okay, Sankeet was saying about the previous answer B, I think so. Diya Ja is also saying B. I think that is all previous answers. What about the new question? Moksh Sharma, no. Option A is not the answer. Moksh, I forgot. Were you plus subscriber? Did you attend my plus courses? So everyone is saying different answers. Let's see. A, B, D, D. Acha. Right. Let me tell you. The correct answer is, who told the correct answer? Divya and Divya told the correct answer. For gluconeogenesis, what do you need? You need pyruvate to oxaloacetate. Okay, oxaloacetate is involved. Oxaloacetate. Oxaloacetate is produced by carboxylation of pyruvate. Okay, oxaloacetate is produced by what? Carboxylation of pyruvate. See, pyruvate carboxylase reaction, which is stimulated by acetyl CoA. Citrate activates acetyl CoA carboxylase, but acetyl CoA carboxylase is going to produce what? Malonyl CoA. Malonyl CoA is involved in fatty acid synthesis. Is this clear? Sankeet, Deepan twins, Moksh, is the answer clear? Excellent, Divyaja. You have given the correct answer. So remember, pyruvate is converted into acetyl CoA by pyruvate dehydrogenase enzyme. Okay, and pyruvate is converted into oxaloacetate by pyruvate carboxylase enzyme. So the pyruvate is usually produced by glycolysis, that is from your glucose. Whereas fatty acids also produce acetyl CoA. So when there is lot of acetyl CoA coming from fatty acids, this acetyl CoA will go and inhibit this pyruvate to acetyl CoA conversion. Whereas this acetyl CoA will go and activate pyruvate carboxylase reaction, because acetyl CoA, it condenses with the oxaloacetate to produce citrate. Right? Acetyl CoA to oxaloacetate, citrate, that is the first step of TCA cycle. Your oxidation of acetyl CoA in the TCA cycle requires oxaloacetate. So that is why acetyl CoA produced from fatty acid, it stimulates the carboxylation of pyruvate so that oxaloacetate can be produced. Is this clear? If you don't understand anything, feel free to ask. That is the whole point of live classes. right? Tell me the answer. What is the mechanism of action of toxin produced by the bacterium shown in this figure? So, what kind of bacterium is shown here? So, I hope this is very easy question. So, what are you seeing? You are seeing Dr. Azam, excellent Dr. Azam, Deepan, Divya, Moksh, Amku Jain, Sankeet. Hmm? So, what is this? Hmm? So, there is a confusion whether it is Clostridium or diphtheria. Hmm? Oh, Dr. Asam, last time also you attended the class. So, I am so glad that you are attending. So, please tell our college juniors to attend the classes. So, I want to reach out to the college. Thank you for attending. So, this is Corynebacterium diphtheria, and the mechanism of action of diphtheria toxin is ADP ribosylation of elongation factor 2. Right? So, this is easy question as many of you are answering nicely. Good. Let's move ahead. Which is not a channelopathy? What are channelopathies? Channelopathies are disorders involving channels, right? Hmm? So there is also known as proteopathy. In Harper, they have mentioned proteopathies. So proteopathies are disorders which are due to defect in protein folding. Okay. So which is not a channelopathy? Many of you are telling it correctly. Excellent, excellent. Nitish, Deepan, Jasmine, Dr. Azam, Purushot, Moksh, Gauranga, Sankeet, Divya. Right. Excellent, excellent. So, all the others are due to some channel. Okay. Tay-Sachs disease is not due to defect in a channel. So, look at this. So, for this periodic paralysis, I have given the table. Later on, you can go through the table. Okay. So, look at the channels. Sodium channel, calcium, potassium, right? So go through it on your own. Hmm? Good guys, nice. So it seems you are aware of all of these. Nice. So here this is gain of function mutation. Here it is loss of function mutation. So I found this table quite informative. And you see all of them are inherited in dominant manner. So no need to worry. So guys, if you have not subscribed for the Unacademy Plus platform, this is the correct time to do. Okay. Tell me next question answer. Pyridoxal phosphate is required for the activity of all of the following enzymes except Nitish, Moksharma. So they are saying it is option D. Right. So glucose 6-phosphate dehydrogenase is what? It is G6PD only. So you are all correct. So all of you answered correctly. G6PD, it requires niacin. Okay. Good, good. Nice. Answer the next question. So this is also a clinically oriented question. So there are a lot of household contacts of patients of tuberculosis. So it's like in India, right? Almost everyone is exposed to tuberculosis. But some develop the disease, some don't develop the disease. So there are a lot of host factors involved in the development of disease. So in this person, as a prophylaxis, they gave INH. INH will cause deficiency of what? Deficiency of pyridoxine, right? Hmm? So it interferes with the action. So to be specific, it interferes with the action of pyridoxine, we say. Clear? So why there is peripheral neuropathy? Because the action of pyridoxine is affected. Both deficiency and toxicity of pyridoxine both leads to peripheral neuropathy. Remember this point also. Okay, good. If pyridoxine deficiency, right? If pyridoxine deficiency is very chronic, then that will lead to niacin deficiency. So pellagra-like manifestation. Okay, Nitish. So, but let's not go that much. Does treatment with rifampicin leads to vitamin D deficiency? Okay, so I have to look at that, Deepan. Okay, right. Next one. So the last option is multiple dehydrogenase deficiency. See, which other vitamin deficiency it causes pins and needle sensation? Dr. Asam, think about that. Neuropathy, not just peripheral neuropathy, it causes neuropathy. Which vitamin deficiency causes neuropathies? Hmm? Okay. What about B12? B12 has neurological manifestations, right? Hmm? Yes, vitamin E. Vitamin E will also cause neuropathies. See, B12, it also causes peripheral neuropathy. Okay. What about B1? Hmm? B1 will also have neurological manifestations. CNS manifestation, na? You are all saying vitamin E, na? Deficiency of vitamin E will also cause CNS manifestation. Excellent. Someone told subacute combined degeneration of cord, right? Hmm? Subacute combined degeneration of cord. Good, good. Mani Megalai told nicely. Good, Mani Megalai. Tamil Anban. Nice, nice. So vitamin E deficiency also causes neurological manifestations. So you have it in vitamin E deficiency. You have even manifestations, right? Retinitis pigmentosa like manifestations. Hmm? Excellent, guys. So proud of you. Hmm. Good, Nitish. Excellent. Nice, nice. So this question, every one of you answered this correctly. So the person is advised to take biotin supplementation, and it is option A. Clearly, you have tomcat urine in the question. Tomcat urine smell. That is due to multiple carboxylase deficiency. Right. Answer this. An overnight person went on high carbohydrate and low fat diet in order to lose body fat? Tell me. Okay, so someone is asking about cabbage order in PKY. No, in PK phenylketonuria, if we don't have cabbage order in phenylketonuria, we have mousy order, musty order, na? Huh? So that order is because of certain metabolites. For example, in phenylketonuria, you have the excretion of your this phenyl acetate, right? This phenyl acetate, that is the phenyl ketone, that smells like this certain compound same present in your this mousy order. Okay, cabbage order, we have boiled cabbage order and cabbage order, there are two. Okay. Hmm. I think cabbage order, I think you will have in tyrosinemia. Okay, am I right? So can someone correct me? Right, what about this? An overnight person, sorry, overweight person went on a high carbohydrate, low fat diet to metabolize excess carbohydrate. Which vitamin you need? Let's see how many of you answered correctly. Again, the answer is thiamine, right? So RDA of thiamine is directly proportional to the intake of carbohydrates because thiamine is required for pyruvate dehydrogenase, alpha ketoglutarate dehydrogenase, and transketolase. So these are the three enzymes involved in carbohydrate metabolism that are thiamine dependent. Right. Which is the only prokaryotic DNA polymerase with 5' to 3' exonuclease activity? What is 5' 3' exonuclease activity? This is involved in DNA repair and RNA primer removal. So there is only one DNA polymerase with 5' 3' exonuclease. 1, 2, 3, they all have 3' to 5' exonuclease. That is your proofreading activity. But Moksh Sharma, let me change the answer. It is DNA polymerase 1 that only has 5' 3' exonuclease activity. Clear? So DNA pol 1 minus this activity, minus 5' 3' exonuclease. What is the name of this? If you remove 5' 3' exonuclease from DNA polymerase 1, you get a fragment that is known as clino fragment. If you remove the 5' 3' exonuclease activity, excellent Dr. Gibson, excellent Dr. Deepan, good Sankit, nice. So let me, someone was asking about urine analysis. Let me open urine analysis. So here I must be having all these smells, odor of urine. Let's quickly see. So I was correct only, but still I think here only These three are given. That is some other PPT which I have taken. Okay, so Deepan is saying, confirming that cabbage order is in tyrosinemia only. Okay, glad Deepan that you confirmed. Good guys. Mahesh Shaini, good evening. So guys, download the Unacademy app also. So in the Unacademy app, we have lot of special classes going on, free classes, and there will be excellent polling feature. So here you are answering in the YouTube comment, right? So there in the poll itself you can answer. So okay, Tamil Anban is saying, what is Kornberg enzyme? Kornberg enzyme is nothing but DNA polymerase 1. DNA polymerase 1 is known as Kornberg. Arthur Kornberg, right? So Arthur Kornberg, okay, Kornberg, he discovered DNA polymerase 1. So that is why it is Kornberg enzyme. So we have name, right? So there is something known as Warburg yellow enzyme, right? Huh? So what is this Warburg yellow enzyme? Hmm? So anyone knows Warburg yellow enzyme? Tell me. See, look at this. I did a quick Google search. Hmm? So these yellow enzymes are actually enzymes that contained vitamin B2. So these are flavoenzymes. People say that Warburg yellow enzyme refers to B2. How can a B2 is a vitamin, right? It cannot be enzyme. So what are Warburg yellow enzymes? These are the enzymes with FAD as a prosthetic group. Okay. But people say that, okay, Warburg yellow enzyme is riboflavin. So that is not correct. Also, people also confuse inverts sugar. So to be precise, invert sugar, it refers to the mixture of sucrose hydrolysis. Okay, sucrose hydrolysis, this mixture, right, that is actually known as invert sugar. Okay, so sucrose itself is not, to be precise, sucrose itself is not invert sugar. Okay, the hydrolysis products, right? So here the change of direction of light will be different. Okay, so that is why, right? So I think we have deviated from the topic. We went into chemistry of carbohydrates. Good. So in next week we will have our live session again, 11 p.m. Okay, so thank you all. Thanks for joining. See you in some other session. Bye bye. Enjoy your weekend. Keep preparing.